Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today my presentation title is Seed Production Principles. Before presentation I would like to introduce myself. I am Vijay Kumar Shrivastav. I have completed M.Sc Agriculture with a specialization in Agronomy from Pant University of Agriculture and Technology, Pantnagar in 1996. So let's move to the presentation. What is seed? Seed is a plant part having living embryo that have a capacity to produce identical plant, and it is used for raising seed crop or commercial crop. Seed is the carrier of new technology. The introduction of quality seeds of new varieties wisely combined with other inputs significantly increases the yield levels. If we will talk about seed, the typical seeds include three basic parts. First one is embryo, second is supply of nutrients for the embryo, and third, seed coat. Seed technology. Seed technology is the discipline of study having to do with seed production, maintenance, quality and preservation. It can be defined as the method through which the genetic purity and physical characteristics of seed could be improved. It involves such activities as varietal development, evaluation and release, seed production, processing, storage and certification. So this is one of the important branch of agriculture by which we can make the necessary development of new demand of lines, varieties, hybrids which can meet the demand of farmers to produce the good quality of seeds and high productivity to feed the population. Now to study the key deliverables of efficient seed production technology. First one is highest order of seed quality in terms of genetic purity, seed vigor and germination. Seed is the primary source for farmers, so we should ensure the good germination of seed, good seed vigor which will result in better crop standing, and it should contain the true characteristics of lines or varieties or hybrids, which is genetic purity. So before supply of the seed to the customers, we have to ensure these parameters. Second, maximization of seed yield per unit area. We have to develop the technology by which we can increase the yields and productivity. Then third, reduction of production cost per kg of seed. We have to develop the tools because of which we can minimize the expenses and return on invest should be higher side. Then fourth, freedom from inert matter, weed seed and other crop seeds as per prescribed standards. The seed should be physically pure and physically purity should be excellent and it should be free of all these. Then fifth, freedom from seed-borne diseases. Whenever we are supplying the seed, it should be free from any contaminants. Otherwise, at the maturity stage, there will be development of diseases which will ultimately reduce the yields. And after that, sixth, timely availability of seed to the customer. Our aim is to produce good seed and it should be available to the customers on time before commencing of season, because of which they can get the profit by cultivating good hybrids and get the profits. Now to study seed production. Production of genetically pure and otherwise good quality pedigree seed is an exacting task requiring high technical skills and comparatively heavy financial investment. Seed production having the strict attention for the maintenance of genetic purity and other qualities of seed. Seed production must be carried out under standardized and well organized conditions. So in case of seed production, we have to follow all precautions related to maintaining the purity. It may be genetic or physical purity, and technical staff should be there for monitoring the activities, and we have to follow all technical guidance and skills which is given by the specialist. Now to study importance of seed production. It is important for multiplication of superior varieties, availability of superior varieties to masses and timely supply, maintenance of genetic purity and supply of high quality seeds. Seeds should have high germination status, seeds having high vigor and viability status, maintaining excellent health status of seed, and securing high crop yields and income level to farmers. So these are the primary importance of seed production. Now to study the characteristics of quality seed. The quality seeds should have high genetic purity, high pure seed percent means high physical purity, high germinability, high seed vigor, higher field establishment. It should be free from pests and diseases. Seeds should have uniformity, like having good shape, size, color according to the specification of variety. Seeds should have high longevity, shelf life, and optimum moisture content for storage and high market value. Now to study the safeguards for maintenance of genetic purity. First one is control of seed source. The seed source should be pure, it should be trusted, and it should be free from any contamination or admixture. Second, preceding crop requirement. The seed should be produced in a described requirement of seed production conditions, whichever is needed prescribed. Third is isolation. The seed should be produced in isolated conditions to avoid any contamination or admixtures. Fourth is roguing of seed fields. Roguing should be done timely and effectively to remove the undesirable plants because of maintaining the genetic purity and physical purity. And fifth, seed certification. The seeds should be passed through all tests and it should be okay and verified and released as a good seed conditions. Then sixth is grow out test. Grow out test is the method of testing of the genetic purity of the seed where the seeds are grown in field condition and the data is recorded of flowering behavior and fruiting structures also to verify its purity. The principles of seed production involve genetic principles and agronomic principles. Genetic principles deal with the purity of seeds and its maintenance, and agronomic principles involve the all practices to be followed for the production of seed and its multiplication by maintaining the genetic purity. What is genetic purity? It is the true to type nature of the seed, like seedling, plant and tree from the seed should resemble its mother in all aspects. This quality character is important for achieving the desired goal of raising of the crop, either yield or for resistance or for the desired quality factors. Now to study the safeguard measures for maintenance of genetic purity. First one is control of seed source and use of approved seed only in seed multiplications. The seed source should be trusted. The seed should be totally pure, free from any contaminations and admixtures. Second, inspection and approval of field prior to planting. The seed production conditions should be verified and checked. In fields where we are going for seed production, the same crop should not be grown in previous seasons and it should be free from any volunteer plant contaminations. Third, providing adequate isolation to prevent contamination. Isolation is one of the important parameters to maintain the genetic purity. In seed production conditions, there should not be same crop grown within isolation distance to avoid any contamination with foreign pollen. Then seed multiplication outside their normal cultivation area. Seed production should be done in isolated fields out of commercial cultivation area, traditional areas, to avoid any contaminations with volunteer plants or side by side grown crops. So it should be totally separate. Then roguing of seed fields at critical stages. We have to follow the roguing compulsorily to maintain the genetic purity, and it should be done on different stages where we can observe the contamination at extreme level. So there are some critical stages which are very important and roguing to be done. After the periodic testing of varieties for genetic purity, we have to check the genetic purity of the seeds on interval basis to maintain the reports and avoid any contaminations and purity status. Then certification of seed crops to maintain genetic purity and quality of seeds. The seeds should be certified and it should go for different testing processes like germination or viability, genetic purity, grow out test, electrophoresis test, etc. to verify its genetic purity as well as physical purity and other status. Then frequent field visits and monitoring during all operations. In seed production, it is the primary responsibility to make the monitoring visits at different stages to avoid any quality issues. Then grow out test. Grow out test refers to the growing of seeds for testing purpose, where we are observing the data on flowering basis and if needed to observe the flowering characteristics and seed fruiting body also. So grow out test is the method for verifying the genetic purity at field conditions. As agronomic principles are related to seed production, we have to select the agroclimatic regions which are suitable for seed production. And in these agroclimatic conditions, there are different factors like photoperiod, temperature, rainfall, humidity, this sun insolation means sunny days, and wind velocity, which are affecting the seed production activities. Like it should be favorable to the crop, like it should not be high extreme temperature where the pollen viability will be affected and growth will be impacted, and the rainfall should be moderate and moderate humidity also, and it should be dry sunny days. So if a crop is meeting the climatic parameters, then we can proceed for seed production in such area. Some climatic factors which can influence the seed production we will discuss here, like excessive dew and rain can cause the hindrance in pollination. Too high temperature cause desiccation means drying of pollen and resulted poor seed set. Hot and dry weather in case of vegetables, legumes fail to seed set effectively and leads to seedless fruits. And vegetables requires cool climate and low humidity for flower and pollination. So here we want to mention that different crops are having the different climatic requirements. So we have to select the geographical area which are suited to those crops. Like in case of rice, the climatic conditions and requirement will be different, while in case of dry crops like pearl millet, bajra, maize, the climatic conditions will be different. So we have to opt for the production of seeds of different crops according to the suitability to the area. For cross-pollinated crops, adequate wind velocity must be required to complete pollination, because in this case we are using two parents, male and female parents, and where the pollen is moving from male parent to female stigma for the crossing purpose. So wind velocity is playing important role for pollen transfer. Oilseed crops can tolerate high temperature, but rise of terminal heat may lead to forced maturity, resulted a small seed size, and so very cold temperature also damage seed quality in early phases of seed maturation. Seed crops for Rabi season must not be grown on such areas where winter rains prevailed, because it may cause seed quality deterioration at the harvesting time and also make harvesting a big problem. Damp and humid weather increase the disease and pest attack on the field as well as a storage site. So here we can say that temperature, wind velocity, rainfall and humidity all are playing important. Now selection of seed plot. When we are going for seed production, the first process is selection of plot. In seed plot, the soil should be fertile and it should be suitable for respective crops. Site should be free from volunteer plants, and preceding crop must not be the same crop, and it should be free from weeds and other crop plants. Site must be free from soil-borne disease and pests, and site must be leveled and having irrigation facilities. So if these parameters are met, we can proceed for seed production activities. Land preparation. Land for seed crop must be well prepared well leveled so that water stagnation must not take place. Good land status enhance good seed germination, field emergence, and stand crop establishment well. Well pulverized seed bed and trace must be picked up and removed before sowing. So, for the good germination and irrigation purpose, the soil should be leveled and it should be well pulverized. Now, selection of seed source. The foundation seed should have quality as per seed standards and it should be from truthful sources. In case of open pollinated OP varieties seed production, we have to use single parent, and in case of hybrid seed production, we are using two parents, male and female parents, A line and R line are used. Varieties which are adapted to the agroclimatic regions, it should have high yielding variety, and varieties with excellent features like it should have good quality, earliness and resistance to diseases and insect pests. Now the process of selection of variety. The seeds of appropriate class must be brought with case memo. Check out the seals and tags are remain intact and bags are not torn. Check out the details mentioned on the label with full assurance. Case memo must be kept with care for further verification and verification of seed source, that it should be from trustful source and no chances of any impurity and illegal sources. So, if we in seed production, the seed source is playing very important role because whole our process is depending upon the seed source. Now to study the prerequisites for a good hybrid. In case of hybrid seed production, we are using two parents, female parents and male parent, which are A and R lines. While in case of OP varieties, we are using only single parent. So hybrid seed production is differ from OP varieties. In case of hybrid seed production, the female should have a stable cytoplasmic genetic male sterility system, and there should be good combining ability of male and female parents. It will result in good seed setting and mating system. So the parents should be suited to each other. After that, adaptability across environment, then perfect synchronization in flowering. As in hybrid seed production, we are using two parents. So for good yield output crossing, there should be matching of flowering, synchronization of flowering. As male and female parents have different behavior, so we have to observe the flowering behaviors, and accordingly we are following the staggered sowing system in hybrid seed production. Staggering means different time sowings of male and female parents depending on their flowering behavior to match the same time flowering, which will result in better outcrossing. After that, height of the male and female parent. As we are using two parents, so the parents characters will be different, and generally if we will say height of male should be higher compared to female parents, because of which the pollen will fall from male parent to the stigma and it will result in better outcrossing. After that, stigma receptivity and pollen dispersal. Female should have good stigma receptivity and male should have good pollen load capacity and dispersal. It should be ease in seed production, and in hybrid seed production there should be high and economical seed yields and high yield of yields on commercial scale crop. Whatever the seed we are producing, it should have characteristics of high yielding in commercial scale. So these are the prerequisites for good hybrid. Isolation is one of the important parameter in seed production which is needed to maintain the genetic purity. Isolation of distance must be maintained as per the requirement and it will vary from crop to crop. Isolation distance is the minimum separation required between two or more varieties of the same species of the crop for the purpose of keeping seed pure. Isolation is of three types. First one is spatial isolation, which refers to the physical distance between two crops of the same species where no one will impact the genetic purity of each other. Means we are maintaining the sufficient distance which meets the isolation parameters. And second is temporal isolation, which is also known as time isolation. It means The isolation gap means flowering gap between two crops where no one is impacting each other. Means if one crop flowering is over, then other crops flowering is start, then it will known as temporal isolation. Means we have to mention that both crops flowering should not match same time. Third is physical barrier or physical isolation, where we are using the different methods to avoid the pollen contaminations. We are using the tarpaulins or some hedges to meet the isolation requirement. First type of isolation is spatial isolation. The spatial separation required between a seed field and other source of genetic and mechanical contamination, especially between varieties of cross pollination. In case of cross-pollinated varieties or hybrid seed production, there is lot of chances of foreign pollen contamination, so the isolation distance should be wider. More the extent of outcrossing, wider the distance. The crops in which there is cross-pollination, we have to increase the isolation distance. Higher the class of seed, wider the distance. Means depending upon the classes of seed, the isolation requirement are differ. In case of breeder seed, as we are maintaining the highest genetic purity, so the distance should be at the high level. While in case of foundation seed, there is different parameter, it will be lesser than breeder seed. While in case of hybrid seed production, the isolation distance will be comparatively low than breeder seed and foundation seed. So it will depends upon the classes of seed. In hybrid seed production, wider the distance than that of variety or inbred pure lines. In case of hybrid seed production, the isolation distance should be wider compared to inbred lines and pure line seed production. This spatial isolations is varying from crop to crop. Like in case of maize, the isolation distance will be nearly 250 meters. In case of pearl millet, the isolation distance will be 250 meters. In case of sorghum also 250 meters. In case of a hybrid seed production, the isolation distance is 100 meters. While in case of sunflower, the isolation distance should be 1 kilometer. If we are going for mustard hybrid seed production, the isolation distance should be 1 kilometer because there are contamination chances because of honeybee. So it will vary from crop to crop. Isolation depend upon the type of seed production. In case of inbred pure line seed production, the isolation distance will be less. While in case of hybrid seed production, as we are using two parents male and female lines and there is cross pollination, so the isolation distance will be wider and it will vary from crop to crop. Second type of isolation is temporal isolations, means time isolation. If distance isolation is not maintained, then temporal or isolation distance must be practices by altering the sowing date by differential planting. Crop of seed production should be sown early or late by a margin of 15 to 20 days depending on parent nature. Then neighboring fields of same or other variety to avoid same time flowering and to prevent entry of foreign pollen in the field of seed production. So this is one of the isolation measures to be followed. The isolation time will vary depending upon the crop. For some crops it may be 15 to 20 days, while in case of some other crops where the flowering is elongated, duration is long, then we have to proceed 20 to 30 days gap also. So that temporal isolation should be maintained in such a way that one crop flowering should not coincide with seed production plot. Third type of isolation is physical barrier isolation. In surrounding of crop of seed production, particularly on bunds, crop of well plant height and densely planted should be grown to prevent entry of foreign pollens in the field of seed production. In case of seed production, when it is difficult to maintain the physical isolation distance and temporal isolation, then we have to opt for physical barrier isolation, which can be maintained through growing height plants or use of tarpaulin also. Physical isolation is also practiced by covering the floral parts or whole plant by which we can avoid the physical contaminants into seed fields, like using paper bags to cover the heads. Now we will study about roguing, which is one of the important practice in seed production. Roguing refers to the act of identifying and removing plants with undesirable characteristics from agricultural or seed production field. Rogues are removed from the field to preserve the quality of the crop being grown. Roguing at all stages of the crop in the field is an essential requirement to maintain the genetic purity of the variety. Sometimes rogue plants are not distinguishable before flowering. Therefore, roguing should be done as early as blooming starts. All doubtful plants should be rogued out. The rogued plant should be removed from the field immediately after roguing and destroyed, as they may survive for a few days and may spread their pollen. So, roguing to be done on compulsory basis to maintain the genetic purity in seed production fields. Roguing is done at various stages of the crop. Like first one is vegetative stage or pre-flowering stage, where the roguing is based on morphological characteristics as height, color of vegetation, leaf size, shape, leaf orientation, or diseased or malformed plants. At this stage, wherever some plants are differing from original plants, it should be rogued out immediately. After that, roguing at flowering stage. At flowering stage, the roguing is based on flowering behaviors like pollen color, petal colors, emergence of panicle characteristics, a fruiting body which is setted, etc. And uprooting of such plants are needed on immediate basis. Flowering stage roguing is equally important in case of hybrid crops. In hybrid seed production, in male sterile line A line, which is female line, a plant of male fertile lines B line is present sometimes. Such B line plants are called as pollen shedders, and it must be rogued out at flowering stage immediate after observation. If it will not be removed, it will contaminate the genetic purity, and again in next season there will be emergence of sterile lines. In male lines R lines, roguing should be done first on priority basis before flowering and crossing, because in hybrid seed production the pollen is transferred from male line to female lines, so male lines should be 100% pure to avoid any contamination issues. Maturity stage roguing is important, and it leads to removal of defective ear heads like off textures, off types, off colored, diseased or malformed. So before harvesting, there should be roguing at maturity stage. In case of cross-pollinated crops, if a beehive is present in the close proximity of the seed farms, it leads to higher seed set as supplementary pollination, and it can cause contamination also if isolation issues are prevailing there. Weeds may increase the chances of admixtures, and weeds plants are the source of disease host. Noxious weeds pose a serious setback to seed purity if not controlled, and weed crop compete with the seed crop for food and space. Grow out test GOT. Genetic purity of any variety is confirmed by carrying out through grow out test GOT, like growing of progenies during off season in the field and electrophoresis. These tests are essential part of seed certification of hybrids and high-valued seeds. Grow out test is conducted to determine genetic purity of a seed lot wherever it is prerequisites for grant of the certificate, and also on the seed lots where a doubt has been arisen about the genetic purity. The grow out test can be complemented by certain related laboratory tests also. In case of grow out test, the seed is grown in field and it is observed the flowering behaviors and reproductive stages to confirm the same true type to the parent. After that, the report is submitted. If it is meeting the same characteristics as parent, it will be okay. Otherwise, the seed will be rejected. In case of high valued crop like vegetables, cotton seeds, sunflower, the GOT is mandatory and it is recommended for testing to verify and certify. While in case of open pollinated crops where the seed value is less and there is no chances of any contaminations and no doubt, in such cases the grow out test is not recommended. Some other tests are electrophoresis. Genetic purity of any variety is confirmed by carrying out through molecular method of protein electrophoresis, where protein bands are run on agarose gel along with the reference band. If both test and reference bands are same, then genetic purity is confirmed. There is another test of ELISA test, which is known as enzyme linked immune sorbate assays, which are being used extensively for the identification of Bt protein in Bt transgenic crop. A sandwich ELISA test is the most preferable immunoassay for the quantification of Bt protein in transgenic cotton plants. So these are the tests which are done to verify the genetic purity and presence of GM intents. Assessment of harvesting time. Harvesting time is one of the important factors that influence the planting value of seeds. The moisture content of seed is an important consideration in deciding the time of harvesting. The harvesting time should be optimum. If we are going for early harvest or late harvest, there will be some problems. In case of early harvest, there will be premature status of the crop. It will cause high number of partially filled and immature seeds with high moisture content and shriveled seeds, and seed quality such as longevity and field emergence will be depressed. If we are going for late harvesting, it will result in shattering of grains, germination even before harvesting during raining season, and breakage during processing. So the harvesting time should be optimum, which should give the proper maturity of crop, and it should be escape from the any rain damage. Now to study about assessing maturity and harvesting stage. Crop can be harvested by assessing the maturity based on physiological maturity and harvesting maturity. First, physiological maturity. Physiological maturity denotes the stage of development when the seed reaches its maximum dry weight and marks the end of seed filling period. It also refers to a development stage after which no further increase in dry matter occurs in the economic part. Crop is considered to be at physiological maturity when the translocation of photosynthesis to the economic part is stopped. Maximum germination and vigor is recorded at physiological maturity stage. Second assessment method is harvest maturity. It occurs 7 days after physiological maturity. The important processes during this period is loss of moisture from the plant. Now threshing. Before the threshing of harvest of seed production plot, threshing floor or thresher must be cleaned thoroughly to avoid any physical impurities and or admixture of seeds of any crop and weeds. Threshing of harvest of seed production plot should be done first, then commercial crop. Whenever you are using the thresher, it should be exclusively for seed production fields and it should be not rotated in commercial crop and seed production. So, because the seed is final produce at threshing stage, we have to take care to avoid any contamination and admixture. So here thresher and threshing floor is needed to be observed and cleaned thoroughly. Now storage of produced seeds. Before storing, seed must be sun dried properly to maintain moisture content of seeds. If seed is dried properly, it will improve the longevity, storage of seeds, it will having the good germination and viability. Some cases where the climatic conditions are not favorable and there is cloudy or rainy weather. In such cases, we can put on other options like using artificial seed dryers, where the seed can be dried immediately and it will save the time and safeguard the seed produce. Packing material used for seed storage plays important role to maintain seeds longevity during storage. Under ambient conditions of room temperature, use of air permeable containers like cotton cloths or gunny bags compared to polythene bags are preferable. Seeds should be stored in dry and cool place. In storage, optimum conditions, particularly temperature and relative humidity, must be maintained. And preventive control measures, again storage pest, also should be taken. We have gone through seed production principles. Now we will go through the key issues which are observed in seed production from beginning to end. Like there are some field related issues like wrong field selection, isolation maintenance, poor crop management, wrong nicking and disease and insect problems. So it should be tackled in an efficient way. After that, there is quality related issues like presence of off types, pollen shaders, volunteers, mechanical mixtures during threshing, harvesting and harvest moistures. So it should be taken care and we should take care while rogging and harvesting process. After that, there are some people related issues like unskilled seed producers and unawareness of technical knowledge. So if we are going for the seed production, there should be training to all persons which are involved in seed production activity. After that, there is production cost issues like procurement prices or cost of cultivation, which will vary from crop to crop. So we have to think about minimizing the expenses and increase the benefits. There are some weather related issues like drought or excess and untimely rains, seed discoloration, rain damage, germination issues, disease and insect problem. So to tackle these issues, we should have some pre-planned activities by which we can escape all these things. There are some operations related issues like sowing time, rogging time, harvesting stage, post-harvest operations, mechanical and cleaning operations like threshing and harvesting, drying and bagging, gunny bags for marking and dispatches. As the seed is our last produce, final produce, so a special care should be taken and we should follow all strategic measures to avoid any contaminations or damage. So these are the issues we have to take care in seed production to maintain the genetic integrity, physical purity and best supply of the seed. So this presentation was all about seed production principles. Hope this will be useful to all of you. Now my presentation ends here. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback for further improvements in next coming presentations. Thank you.